Yeah, broken. That's what you're saying. Yeah. Chair's broken. You are in ship. You are in ship.
Just approach it, but before we go into that, I send you an email with the PI value. Some people had questions about the PI of amino acids that have side chains. Again, want to spend 1 or 2 minutes on that. The confusion comes from your extra knowledge of organic chemistry. Because you have that extra knowledge, you know that anything can act as an acid. Alkyl groups can act as an acid. They have extremely high pK of about 60, but that's not really relevant in biochemistry. In the same way, alcohols, alkyl alcohols, also can be acids. They have a pK of around 15, and that is also not relevant for us. So most of the time, we don't really talk about alcohols as an acid. The only time alcohols can be significant acids are phenols, that means they have an aromatic ring. And then the pK of that is around maybe 10, and that is in our range as you see from the pK of the various groups that we are discussing in amino acids. So only tyrosine, which has a aromatic group, is listed in the list of pK values. Others are not. Serine, toluene, they are not. And SH, H2S is a stronger acid than H2O, that you already know because sulfur is a bigger size, and that's why SH also is a stronger acid, and that has a pK well within the range around 6 or 7, and so that also is listed there. If you want to determine the pI of any amino acid that has a side chain, you have to first decide is the side chain acidic or basic. It is very easy to do that. If it has nitrogen, that is basic. So for example, lysine, arginine, and histidine, they have nitrogen in them, so they are basic, whereas the tyrosine or cysteine, which have OH or SH, they are acidic. Whenever you have, and of course, aspartate and glutamate, they are strongly, they have a carboxyl groups, they are obviously acidic. So if you have two acidic groups in any amino acid, whether it is the tyrosine or not, whether it is cysteine or aspartate or glutamate, all you do for the pK is to take the average of the first two pK values. Go from the lowest to the highest, and the first two pK values take the average of that. That is the PI of that amino acid. Yes?
But for example, if we're determining the PI value, are we going to be given the pH values of the side chains? And
you don't need to memorize anything.
Okay.
So, and once again, if there are any calculations, there will be calculations, but there will be no calculations that require a calculator. So, if I give you such a problem, I will basically adjust those pH values and make it a hypothetical amino acid and make it a whole number pK so that you can easily do addition, subtraction, or average, divide by 2 that you don't need a calculator for. All right, so those pK values given will not be real, but will be made easier by making them whole numbers. So you will not need a calculator for any calculation. And you don't need to memorize any pK values. So if you have two acidic groups, you take the average of the first two pK values, pK from the lowest to the, going from the lowest to the highest. For example, if you have the alpha carboxyl group plus, let's say, the aspartate carboxyl group or the glutamate. If you have two amino groups, then you take the average of the two highest ones. Like lysine, arginine, you take the amino group and the alpha amino group. Yeah, and right in the middle falls histidine. As I explained, histidine is an important amino acid. It has a pK right around neutral, and that gives it special properties. So for the PI of histidine, you take the average of the two top pK values. And because it is amino group, you take the average of the alpha amino group and the histidine nitrogen. Alright, so very easy. If it has a nitrogen, it's a base. If it has other things like carboxyl group, OH, and it is listed in the list of pK values, then it is acidic. Because other ones will be not acidic enough for our consideration. Alright, so going to the next part of the chapter, 3D structure of proteins. And 3D structure from your organic chemistry, you know, it depends on conformations. You studied conformations and you know that there can be numerous conformations. Just for ethane, you saw it can be staggered eclipse conformations, it goes back and forth between them. Instead of ethane, if you have butane, you get more conformations. You have more carbons, you have more conformations. For an amino acid or a protein, amino acid itself has several carbons and a protein has hundreds of carbons. So how many conformations are possible for a protein molecule? It is really infinite number of conformations. As the number of atoms increase, the number of conformations increase exponentially. And for proteins, there can be unlimited number of conformations. Out of all those, there is only one that the protein exists in. That is called the native structure. So out of trillions of conformations, only one of them is the most stable, obviously. And that is the structure in which the protein exists, and that is called the native conformation. If it is not the native conformation or native structure, then it is called denatured structure. If it loses activity, there may be a slight variation in some conformation that still maintains the activity. Then it is still in some variation of the native, which doesn't exist much. But most other conformations will lose activity. The protein will lose activity, and then it is called denatured. So there is nothing called half denatured or full denatured. Either it is denatured, it has activity, or it doesn't have activity. You can have 50% denaturation. That means 50% of the molecules are denatured. If any time a book says this is 50% denatured, it is not talking about one molecule being half denatured and half not. One molecule, if it is denatured, it is 100% denatured or 0% denatured because it is either active or not active. But you may have a collection of molecules because there are, of course, Avogadro number of molecules. Some of them may be denatured, some of them may not be. That's what gives rise to different percentage of denatured proteins. So in order to understand that, we need to then look at the conformation, and that arises from the peptide bond. So this is the peptide bond or amide bond, and you can draw a resonance structure of this. This nitrogen has a pair of electrons, so if we draw a resonance structure, so this is the resonance structure of the peptide bond, and as you see, the peptide bond has a double bond between the carbon and the nitrogen. And if you want to take a resonance hybrid structure, which one is more present? This is about 40%. So that means all peptide bonds have about 40% double bond character, and so it gives properties of a double bond. And what is the property of a double bond? One property you learned that it can have cis and trans. One property you know about, and the source of cis and trans is the fact that there is no free rotation around double bonds. You can have free rotations around single bonds, but not around double bonds. So there is no free rotation around this. And just like for any alkene, you have a cis and trans, so peptide bonds will also have cis and trans. So that's one factor. And the other thing that you know about, let me draw this. So because it is a alkene, all these atoms, 1, 2, 3, 4, 5, 6, all these 6 atoms are in one plane. That is the property of any alkene. And you cannot break that plane. In other words, you cannot have rotation around those planes. But then there will be, the next carbon is the C alpha carbon, which has the R group, and of course a hydrogen. And then you have another peptide bond. So in this one also, you have all 6 of these in one plane. Alright, so they have to be in one plane. It says that that's according to the alkene stereochemistry. So this has to be in one plane, this has to be in one plane. It doesn't say that these 2 planes have to be the same. They can be different planes, but each of them is one plane. So if you have 2 peptide bonds like this, so this is a peptide bond and there are these 4 atoms at the 4 corners. So all these 4, including the 2 in the middle, have to be in one plane, and this has to be in one plane. But these 2 planes don't have to be the same. It can be like this, or like this. Right? So there can be rotation of the 2 planes around the C alpha carbon. And so how much you rotate? It can rotate anywhere from here to here, 360 degrees for this, 360 degrees for this. So each of them can rotate by 360 degrees. And you give you want to differentiate between the 2 planes. So the one that has the carbon-carbon and the carbon-nitrogen. This is the easy way to remember. So the angles of rotation around this one is called the psi angle. If you want to remember, psi for C, it's spelled psi, of course. And this angle of rotation around the carbon-nitrogen is called the phi angle. And each of them can be 360 degrees because the planes can rotate and make one full turn. Now, as it rotates, these different groups will come. Of course, if you're rotating, then this oxygen will come here. The R will have interaction between that oxygen. This hydrogen of the nitrogen can come on this side as it rotates, and that R group can have interaction with that hydrogen. So if you work with a model, you find that as you rotate, some of the atoms come too close to each other. That means there will be steric hindrance between them. And so that angle of rotation is not allowed. So working with models, you can figure out which angles are allowed and not allowed. That's not an easy job because it's a mathematical thing. You have to calculate what is the energy of interaction between the different groups for every angle of rotation. So this is if you want to plot a graph of that, then this will have 4 quadrants, because let's say you are rotating around in clockwise, then plus rotation will be 0 to 180 degrees. Minus rotation will be 0 to 180 degrees on this side. Same for the phi rotation, 0 to 180 and 0 to 180 on the other side. So there will be 4 quadrants, and you have to do calculations. For example, let's say here, is this angle allowed? So this angle, how much is this angle? You have rotated around this one by this much, and around this one by this much. So is that angle allowed or not? And that way you do calculations for each of these points in the whole 4 quadrants. How easy or difficult calculation is this? That must be a very difficult calculation for doing each of them. And all this was done before the days of computer, before the days of even a calculator. Alright, so all these angles were calculated, and then we know which angles are allowed and which angles are not allowed. So this way you have dots like this coming up, whichever angle you try, this is allowed. any white area, those angles are not allowed. Yes?
Allowed or not allowed to what?
For steric hindrance. If there is steric hindrance, as you are rotating, two atoms come close to each other, then that is not allowed. So the atomic radius of one, if it overlaps with the atomic radius of another, then there will be steric hindrance. So that angle is not allowed for rotation. Remember, these conformations for any molecule, they happen at random. They keep on moving, right? But then some, for example, you learn just for ethane, you know the staggered is more stable than the eclipse. So this way there are millions of conformations. Some of them are more stable than others. Some of them are not stable at all, so they don't happen. So those angles are called not allowed. So proteins will not exist in those conformations. So all these calculations were done even before there were computers or calculators. And this kind of a plot where you are showing what is allowed and what is not allowed for the protein, that is called the Ramachandran plot. So Ramachandran plot shows which angles in the four quadrants for the rotation around the CN and the CC bond are allowed or not allowed. By allowed or not allowed, it means whether the protein exists in that conformation or not. Nowadays, in the age of computers, all you do is feed in the sequence of the protein, and the computer will immediately give you what is allowed and not allowed. So this way, if you, I emailed you the Ramachandran plot of this protein. Polyalanine. Now this you have to do, this calculation you have to do for every protein. It's a different calculation, depends on the sequence, depends on the R group. Remember, the whole thing is happening because of steric hindrance with the R group and the other groups. So R group, there are 20 different R groups for the 20 amino acids, so you have to do it for every amino acid. So if this was done for polyalanine, that means all of them are a methyl group, and they came up with a diagram something like this. This is somewhere in this area it is allowed, somewhere in this area it is allowed, somewhere in this area it is allowed. Most of the angles are actually not allowed. That means in those angles there will be steric hindrance. So it is logical, if this is the question in the exam, if this is the diagram for the polyalanine, how will the diagram for polyglycine look like? Will it have more allowed angles or less allowed angles? Yes?
More.
More, because the R group is only a hydrogen. There will be less steric hindrance. So that means there will be less of the white area and more of the dark area, whatever color you want to put in there. Or if you are doing it for tryptophan, which has a big R group, then there will be even less allowed angles. All right, so if you take one peptide bond and let's say you rotate it based on what is allowed or not. Let's say this angle is allowed and this is possible. What about the next peptide bond? That also can be a different amino acid and that will also have an allowed angle. If the next one also rotates like this by the same amount, then that is allowed. And if the next one also rotates by the same amount, what is the structure you are going to get? You are going to get a helical structure because every peptide bond is rotating by the same angle. And that's how you get a helical structure, and that gives rise to a three-dimensional structure of protein. So there are three levels of structures in the protein, three types of three-dimensional structure. One is called the alpha helix, and then beta sheet, and random coil. So let's start with the random coil, which the name says it is random, but it is not really random, as we already saw that the angles that are not allowed will not be allowed. It doesn't exist in that. So random coil means one peptide bond has a certain angle, the next peptide bond is independent of the first one, it can again have any of the allowed angles. Right? So if this angle is allowed, let's say 20 degrees to the right, and the next one 40 degrees to the right is also allowed, the next one has that angle, the next one has maybe this angle, so that gives rise to a random structure. Every peptide bond has a different angle. It will never violate the Ramachandran plot. It will always follow those allowed angles, but those allowed angles may be many. And it can choose any one of those. And if that happens, that gives what is called a random coil. So once again, it is not absolutely random. It has to still follow those angles. But it can be one peptide bond is independent of the other peptide bond. However, if they have the same angles, then it gives rise to these two structures, alpha helix and beta sheet. And so the alpha helical structure, which of course is drawn in the handout, but I'm not going to draw it again, so it will look like this. So for any helical structure, for example, DNA is a helix, there are a couple of questions that automatically come to mind. One is, which direction is the helix? Remember, helix can be right-handed or left-handed, right? So the alpha helix is a right-handed helix. The next question that comes to mind is, how many amino acids make one complete turn? So if you are going from, let's say from this point to this point, so how many amino acids are there to come to this point? So number of amino acids per turn. And then what is the distance between these? You take any identical point on the helix, like this one to this one, or this point to this point. So that will be, that's called the pitch. And the pitch for the alpha helix is about 5.4 nanometers. And the number of amino acids per turn is 3.6. So a simple calculation, how much is the helix rising per amino acid? And that is called the rise, because this is a helix, so it's obviously going up. And for every amino acid, it goes up a certain distance. And that, of course, will be the pitch divided by 3.6, which is the number of amino acid. So that's called the rise. And that's about 0.15 nanometer per turn. So once again, these numbers are not important. Just know that if it's a helix, these are the properties that it will have. Every helix will have these properties. The numbers may be different. Now the question is, once again, the helix happens because every peptide bond has the same angle of rotation. But why does it have the same? The second one can have any of the allowed angles. It can be this way or this way. Why does it have to be the same? So obviously there must be some thermodynamic property that is allowing it to have the same angle repeated. So what contributes to the stability of the alpha helix? What is forcing each peptide bond to have the same angle of rotation? Because that gives it extra stability. So what is that stability? So let's say this is a helical structure. Somewhere here there is a carboxyl group, for the ketone for the peptide bond. And when it has this 3.6 turn per 3.6 bases per turn, it so happens that right below it is an H. and that gives hydrogen bonding between these. So if you have a carboxyl group here, and right below that will be a NH that forms hydrogen bond. And that also tells you why it is 3.6, why not 4? If it was 4, then a carboxyl group will have another carboxyl group right below it. Or amino group will have another amino group right below it. So there won't be any hydrogen bonding. So it is slightly less than 4, so that every amino acid, every peptide bond will have its CO directly above or below a NH of another peptide bond. That means another 4th peptide bond. And that's what makes the alpha helix a stable structure. That is what makes it have the same angle repeatedly and give this kind of a structure. Yes.
Is this interaction between R groups of amino acids? Or is
No, this is between the CO and NH. So CO of one peptide bond and the NH of the 4th amino acid from that. And that happens to line up below or above each other, and that's why it is stable. And that's why each peptide bond is having those particular angles. Yes?
Sorry, which part was the rise again?
The rise is the amount of increase per amino acid, so from here to here. So if there are 3.6 of them, then the peak divided by 3.6 will be the rise. All right, so in this alpha helix, where are the R groups? You mentioned about the R groups. The R groups are all pointing outwards. So here is the R group, here is the R group. So R groups are all pointing outwards from the helix. So if you look at the helix, the R groups are all out. Now, the R groups, again, remember the Ramachandran plot. If the R group is big, it will have less allowed angles. And so in the alpha helix also, if you have a very big R group, then it is not favored in the alpha helix. The alpha helix is favored for amino acids that have small R groups, like alanine. So you can take all the 20 amino acids and make some sort of a table, like which ones are more favored in the alpha helix. Remember, a protein has hundreds of amino acids in a sequence. And if all the amino acids in, let's say, 20 amino acids next to each other have amino acid that is favored for a alpha helix, then it will form an alpha helix. If you have a region in which there may be 5 alanines and 15 tyrosine and tryptophan and glycine, they are all big groups, then that region will not form an alpha helix because those amino acids are not favored in alpha helix. If you have one of them that is big, but remaining 19 are okay, or 2 of them are big, 18 are okay, then still it may be alpha helix. So you can make a table of which is more favored to be an alpha helix. In fact, with the aid of computers, you just feed in the sequence and it will automatically give a structure. Even though that structure hasn't been tested or experimentally determined, the computer can say this region looks like an alpha helix because all the amino acids are favored in an alpha helix.
So most of them are favored in the alpha helix?
No, the small ones.
Right.
Small R groups.
So if it's mostly small hard groups, like the one tryptophan, it will still form a helix?
It will probably form an helix. So there may be, out of 20, maybe a couple of them maybe.
And the small ones are like alanine.
Alanine, leucine, isoleucine stuff, those are the smaller. Serine is another one. Cysteine is another one.
What did you say before cysteine?
Serine, cysteine, those are small ones. Then the big ones are aspartate, lysine, arginine, tryptophan. Those are big R groups. So you don't usually see them in an alpha helix. Now, there are 2 amino acids that are absolutely not present in an alpha helix. And when that kind of statement is made that this is totally different for all other amino acids in terms of structure, you can immediately point to one amino acid. If you look in the list of all the amino acids, there is one that stands out to be totally different. Anyone knows which one is that? Yes. Proline. What is special about proline? It has a 5-membered ring. So the R group is basically a 5-membered ring like this. And the nitrogen is part of that ring. So that means the nitrogen is a secondary nitrogen, it's not a primary nitrogen. All other amines are primary, this is the secondary one. And secondly, the nitrogen is part of a ring, and so there cannot be sufficient rotations around this. Every time it rotates, the whole ring has to rotate with it. And so there is hindrance for rotation, and proline is usually the odd protein out whenever it comes to any deviation in the structure of any protein. It is usually, you can blame it on the proline. So proline is called a alpha helix breaker. So let's say again, nowadays everything is done on the computer, and you have a sequence of amino acids, and the computer immediately says the helix ends here. So you have these amino acids one after another, and the computer identifies that from here to here it forms a helix. Why not the next one? Next one can be a tryptophan, but that doesn't mean it. The tryptophan can be tolerated. It's not favored, but still tolerated if the next ones are also favored. So if this is a tryptophan, but next one is an alanine, and next one is a serine. So there are favored ones outside this, so this alpha helix will continue. But if this is a proline, then definitely the alpha helix ends there, because proline is an alpha helix breaker. Yes?
Professor, do you mind writing a little bigger, please?
So, so any of these amino acids here, if they are allowed, if this amino acid is not allowed, whereas these ones are allowed, it may continue to be an alpha helix from all the way from here to here. But anyway, there is a proline, the alpha helix must end there, because proline cannot have any of those fixed rotations, and the alpha helix ends there. So from here to here, it may be an alpha helix. Next, from here to here, maybe it doesn't have any trend, so it can be a random coil, and then from here to here again it may be a alpha helix or a beta sheet. So depending on whether those angles are allowed or not, but anytime you have a proline, that is a alpha helix breaker. And there is another reason why proline is a alpha helix breaker. So one of them is that it is a ring, and then anyone knows what is the second reason, which is even more important than that ring structure. Yes? Yes, what? The angle is because of the ring, yes. The other is, what is the problem with proline? Because it is a secondary amine, and when it forms a peptide bond, it doesn't have any hydrogen left. Remember, if it is a primary amine, in a peptide bond, the nitrogen still has one hydrogen. But if it is a secondary amine, and it forms a peptide bond, it doesn't have a hydrogen left. So if it doesn't have a hydrogen left, then this hydrogen bonding cannot happen. And that is the basis of the alpha helix. Alpha helix is stable because of those hydrogen bonds. If the proline cannot participate in hydrogen bonding, then of course the alpha helix ends there. So proline is the alpha helix breaker for two reasons. One is that the ring prevents its rotations, it has less allowed angles, and secondly, it is a secondary amine that does not have a hydrogen in the peptide bond, and so it cannot form hydrogen bond. There are two amino acids that are alpha helix breakers. One is proline, the other is glycine. Now you may say, oh, why glycine? Glycine should be the most favored one because it is the smallest amino acid. It has only a hydrogen. If alanine is favored, why not glycine? But glycine is not favored in a alpha helix for that very reason, that it is too small. What happens if an amino acid is too small? It has too many allowed angles. That means it has high entropy. It doesn't want to stay restricted into one particular angle. Remember, in thermodynamics, you are talking not just about the energy of interaction, not just the steric hindrance between the groups. Energy is always energy plus the entropy, right? H delta H minus T delta S. So the entropy is also important. And glycine has such a high entropy that restricting it into a particular angle makes it unstable. And for that reason, glycine is also an alpha helix breaker, and that's how the computer predicts that the alpha helix is emptier. If it is a proline or a glycine, the alpha helix ends at that point, and then it may resume another alpha helix after a number of amino acids.
What does high entropy do?
In general chemistry, you studied the delta G. So whether a process happens or not, whether it's a reaction or a process, rotation, that is also a process. Whether that will happen or not depends on the delta G. Delta G must be negative, right? And that is equal to the delta H minus T delta S, which is the entropy. If entropy is very large, then it may not happen because it will be more stable otherwise. So glycine has a high entropy, which means it doesn't want to be restricted into one angle, and it has low entropy. One angle means it has low entropy. If it can move in all possible angles, then it has high entropy, and glycine is able to do that, and that's why glycine doesn't fit into the alpha helix. All right, so the other type of secondary structure is, so what we are talking about here, secondary structure, I will explain that in a minute. So the other type of structure is the beta sheet, in which the protein or the peptide strand is fully extended. So it looks like this, as you would draw an alkane chain in organic chemistry. So that means it is again following the same angle repeatedly. And in this, you can put anywhere, let's say you put a CO, N8, then this is the alpha group, and then CO. Then C alpha. So this way it extends like this. All right. So what is the stability of this? Why would it repeat the same angle again and again to give the structure? There is no stability by intra beta sheet hydrogen bonding. Unlike in alpha helix, where the fourth amino acid had hydrogen bonding, and that's why it had that structure. The beta sheet doesn't have that. This will never have a hydrogen bonding this or with this one. So there is no stabilization of a beta strand. So this is called a beta strand, and a beta strand doesn't exist in a protein. What does exist is a beta sheet. So if you have another beta strand next to it, like this, and that's like confident. The CO, the NH should come close to CO. So let's say there is a CO here, like this. Then there is a hydrogen bonding between the strands. In the same way, you can have another one like this, and there can be hydrogen bonding here, there can be hydrogen bonding here. So there is intra-strand hydrogen bonding, and that's what gives rise to what is called a beta sheet. So a beta strand doesn't exist by itself. There has to be a couple of beta strands next to each other, and that stabilizes the structure, and that is called a beta sheet. And usually it is maybe 4 or 5 or 6 beta strands, one after another, that hydrogen bonds to each other, and that makes a beta sheet. So remember that this protein is just one straight line, right? One protein means you start from the N terminus and goes to the C terminus. It's one sequence of amino acid. So how does 3 or 4 or 5 or 6 of these come together? So in the three-dimensional structure, something like this, there may be a beta strand from here to here, involving a couple of amino acids. And then there may be a beta strand from here to here. And there may be another beta strand from here to here. So this way, they are coming together. They are not next to each other in the sequence. In the sequence, they may be far away. So there may be a beta strand forming from here to here, another one forms from here to here, another forms here to here. But in the three-dimensional structure, this, and this come close to each other to form a beta sheet. So these beta sheets can be of two types, or beta strands can interact with each other in two different ways. They can be parallel or anti-parallel. So the one that I've drawn here is actually anti-parallel. So how do you decide it's parallel and anti-parallel? If you look at in one strand, and if it goes in this direction from the C to the N, and then in the other direction it is actually C to N in this direction. So remember proteins have a direction, right? There is only one strand, so you go from the N terminus to the C terminus. So let's say this is the direction of the protein, and then it comes back here like this. So this is the N, this is the C, and then again you are coming like this. So this is the N, this is the C. And then again it continues like this. So all these are in the same direction. If they are in the same direction, then it is parallel. If they are in different directions, then it is anti-parallel. So for example, if you have one region of this from N to C, and then it goes like this, and then there is another one, this is N to C. And then it may go, and this may be again from here, N to C this direction, and then N to C in this direction. So it's going in the opposite direction this way. So N to C this way, N to C this way, then this way, then this way. So it is in the opposite direction, and that is anti-parallel. So beta sheets can be anti-parallel or parallel, and anti-parallel is more stable than the parallel ones. I send you the structure of this in the email. This is also in the book. Everything is actually in the book. Or you can, these are basic biochemistry. If you don't want to spend money buying the book, you can buy any book. This information is always there in any book. You can get hold of it. So the anti-parallel is more stable than the parallel one, because if you look at the hydrogen bonding between them, in this one, There's a hydrogen bonding here and then a hydrogen bonding here. These bonds are parallel to each other. And this is parallel to this one. So every hydrogen bond is parallel to each other, which happens because the number of atoms between this CO and the next NH are fixed throughout all the strands. And that's why every amino acid, every carboxyl group falls right below an amino group, and so you have hydrogen bonding in the same direction. If it is parallel, so this is anti-parallel. If it is parallel hydrogen bonding or beta strand, then so one hydrogen bonding will be like this, and the next hydrogen bonding will be 1, 2, 3, 4. Will be this way. So if this is a NH and this is a CO, this will hydrogen bond to a CO. So the hydrogen bonds are not in the same direction, and that happens because if you draw it in this manner, you will find that the number of atoms between this and this is 1 here, and between this and this is 1, 2, 3 here. In one strand, the number of atoms between two hydrogen bonds is more than in the other strand. And that's why each hydrogen bond is at an angle, whereas here, the number of atoms between two hydrogen bonds is same throughout, and that's why you have parallel hydrogen bonding. So parallel looking hydrogen bonding happens in anti-parallel beta sheets. All right, and unparalleled or not parallel hydrogen bonding happens in parallel beta sheets.
Was that one that you drew just now, is that parallel?
That is a parallel, yes. That is a parallel beta sheet that has hydrogen bonds not parallel. There is little confusion, but that's what it is. If it is anti-parallel, it is more stable because the hydrogen bonds are parallel to each other.
Why is
Alright.
that one anti?
Parallel. This is the anti-parallel
Like why?
beta sheet.
Why?
If you look in the direction, I've not written the carbon nitrogen, but if you this is how it happens. This is one single chain of amino acids. So one of them goes in this direction, and then if the next one, which I erased, if the next one goes in this direction, so let's draw that again. So N to C, that is in this direction, and then the next one is in this direction. So from left to right, and this is from right to left. And if they form hydrogen bonds, that is
That molecule you drew in the middle, we can't tell if it's anti-parallel or not because we don't have the sequence of the other sheet.
That's right. It depends on the sequence and the 3 dimension. I'm drawing in 2 dimension, but this strand may be somewhere here because the whole thing is a globular structure in 3 dimension. So this one is anti-parallel. This is parallel, and that's because if this strand goes in N to C, then there is a stretch of amino acids that don't participate in that beta strand. And then after a couple of amino acids, then you find another stretch of amino acids that can form hydrogen bond with these, and so that's how it becomes parallel. So that's how it becomes parallel in the same direction, which means there is an intervening length of amino acids that is in 3 dimensions. It may be pointing in this direction or pointing back. And that's how these two come close to each other. All right, clear? Yeah, drawing a three-dimensional object in two-dimension requires some imagination. So we always have to think that this part is not in the plane of the board as written. Alright, so I'm going to erase all this. Alright, so there are three proteins specifically. There are of course thousands of proteins, but out of those three are discussed in the book. One of them is collagen, which is somehow in this chapter. The other one is hemoglobin, which is in the next chapter. So this is collagen discussed here because it has an interesting structure and a sequence. I mentioned that an alpha helix, proline and glycine are not present in an alpha helix because of the different reasons that I explained. But collagen has only those ones. It is a helix, it is not an alpha helix, it just goes in the opposite direction, and it is made up of only glycine and proline. So the formula of collagen is glycine, proline, and hydroxyproline repeated many times. Many times means there are usually 1,000 amino acids or so in the collagen, and so this sequence will be repeated again and again. There can be a minor variation in that. Sometimes it is if not a proline, it may be a glycine or slight other variations that gives rise to 16 different collagens. There is not just one type, there are many types of collagens, and they differ from each other in minor variations from this. But in mostly it will be glycine, proline, hydroxyproline, and these glycine and proline, as you know, are not present in alpha helix, and these are the only ones that are present in a collagen bond. Now, hydroxyproline, what is hydroxyproline? One of the carbons, proline, of course, remember it's a 5-membered ring, and one of those carbons has a OH. One of those carbons means it can be C2 or C3. Again, that is a variation in the collagen structure. The OH can be a 2 and 3. But wherever it is, the hydroxyproline is not one of the 20 amino acids. So if you want to make collagen, you cannot really eat hydroxyproline. That's not going to do you anything. The hydroxyproline is not one of the 20 amino acids, so it will not be used to make collagen. So you cannot just have hydroxyproline from outside in your diet and expect to make collagen, because that hydroxyproline is made after the collagen strand is made. So first glycine, proline, this polymer is made. So first it is made with this, and then every second proline is oxidized to introduce that hydroxyproline. So what will be the name of that enzyme? Enzymes are usually named based on what they do. So if you think what is this doing, it is putting a hydroxyproline on, or putting a hydroxyl group on a proline. So this will be called prolyl hydroxylase. So this one is converted to this by the enzyme prolyl hydroxylase. So basically then it is a oxidation reaction where atmospheric oxygen is used to oxidize the proline to form a hydroxy group. And there it is, yes.
When did you say that the
Hydroxyproline.
when did you say that that was made?
After the protein is made. Protein is made by in the ribosomes, depending on the DNA sequence, and that is converted to mRNA. Then, based on the mRNA sequence, protein is made, which we are not going to study. We may have studied that in some course, molecular biology. So it is made based on the proteins, on the DNA sequence and RNA sequence, where the sequence is that of protein. So it is a sequence of the DNA says glycine, proline, protein. This is what the protein should be, and that's what is made. And then these alternate prolines are hydroxylated, and again, all prolines may not be hydroxylated. That's how you get different types of collagen, depending on what the function is. So what is the function of collagen? It is one of the most available proteins throughout the body. Most of the protein you see is not by weight, but by function and by number is collagen. Because it has functions like it forms the walls of the blood vessels. It forms rope-like structure like the tendons. It has a layer. It is loosely woven collagen under the skin. So different forms of collagen are there with many different functions. So which is the most available protein throughout the body. And they will differ from each other based on slight variations in this form. So one place where you find collagen is in the blood vessels. That's what make your blood vessels. Besides the cells, of course, cells are endothelial cells, but then the strength of the blood vessel is decided by collagen. So the way the collagen was discovered was by the disease called scurvy. Everyone knows about scurvy. That is related to what? How was it discovered? Vitamin C. So people, sailors who were out in the sea for months, they suffered from scurvy because of lack of vitamin C. And what is scurvy? What are the symptoms of scurvy? The scurvy symptom is basically bleeding. So any small cut, you bleed. I mean, very easily you bleed. For example, the gums. The teeth have gums and they have collagen. So any part of the body that multiplies more rapidly will be affected by lack of collagen. Not just lack of collagen, but the right collagen. The structure is there, the gene is there, so obviously the person is making the collagen, but then it is not converting the prolines to hydroxyprotein. And that reaction, prolyl hydroxylase, requires vitamin C. Vitamin C is a reducing agent. The name vitamin, which you have studied in biology, is it has a definition. Vitamin C doesn't really fall in that definition. So it's hard to say. Some people don't even call it a vitamin, because a vitamin is supposed to be used in very small amount, and you don't make it in your body. That's why it has to be present in diet. Vitamin C is one of the vitamins that you actually need in large amounts, not just small amounts. In fact, Linus Pauling, who got the Nobel Prize, his advice was you should take mega doses of vitamin C every day. Not that everyone agrees with him, but he was a Nobel laureate, and that was his opinion. So vitamin C, what does it do? This is what it does. It keeps the prolyl hydroxylase active. How it does that wasn't understood for a long time, but now we know that this is because the prolyl hydroxylase has the iron in its active site, which must be present as the ferrocyanide, that means Fe2+. And because any enzyme that works with oxygen, this is an enzyme that works with oxygen, it is oxidizing the protein. So anything that works with oxygen will have a tendency to oxidize itself. Not every time, but occasionally it will get oxidized. So if this iron, Fe2+, converts to ferric iron, then the enzyme becomes inactive. So occasionally the enzyme prolyl hydroxylase will become inactive because the iron converts to ferric, and you must have something to convert it back to ferrous. That means you need a reducing agent. And the reducing agent in this case is vitamin C. And that's why you need vitamin C for scurvy, which basically the symptoms are bleeding gums like that. And any part that multiplies more rapidly will be affected. All right, so how does this, as I said, proline and lysine don't exist in alpha helix, but they are in collagen for two reasons. Basically one reason. It's the hydrogen bonding between the strands of the collagen. So there are three collagens that form collagen strands that form a helix, and this is a left-handed helix, just the opposite of the alpha helix. I may not be drawing it correctly, you need a different color. So three of them are together forming a collagen fiber. And these stay together because of hydrogen bonding between the hydroxyl proteins. Remember, hydroxyl groups form hydrogen bonds. So one OH forms a hydrogen bond with another OH of the next strand, and that's how you form a strong collagen structure. It can be a very strong structure because of the tendons. It may not be a strong structure. For example, under the skin it is more loose, so it may have more substituents besides hydroxyproline. It may have a lysine. So there are slight variations in the formula, and that gives rise to the different types of collagen. Yes?
Three helixes form collagen.
Collagen always has three helices, yes. And the three helices are held together by hydrogen bonding between the hydroxyprotein. So this is a intramolecular hydrogen bonding, not intermolecular hydrogen bonding as in alpha helix, where hydrogen bonding are within the same molecule. Anyone had a question here? Yes.
So the collagen without the hydroxylated prolines, are those weaker collagens?
Those are weaker.
Okay.
Now, without means it has a proline. That's how the gene makes it. It makes the proline in this place.
Right,
So
so
it's not
not
hydroxylated, that makes it a weaker collagen, and that's how it results in bleeding. So proline, as I mentioned, is always a very weird amino acid. Another feature of proline that is important, which I forgot to mention, is the trans structure of a peptide bond. So remember, if you draw peptide bonds, there are double bond characters. So this is a double bond character here, and this is the C alpha, and then a double bond character in this one. So every peptide bond has a double bond character, and that means one property of alkene is that it's more stable than the trans. So if this is C alpha, the R group here will be pointing in this direction, and then this is the C alpha, R group is in this direction, and the next one will be in this direction. So all the R groups alternate up and down. That is obvious because they have double bond character and alkenes are more stable in trans. So that's how the whole protein structure is. The R groups must be in opposite direction. Except for one amino acid. And which amino acid is that? It is always proline. The most weird one is always proline. Now proline also is more stable in the trans. Not that it is more stable in cis, it is more stable in trans. But the difference between the stability of cis and trans is less than for other amino acids. So 90% of the time, it is trans. 10% of the prolines in any protein can be cis. So remember, these percentages depend on the relative energies of the cis and trans. If the energy difference is large, then it will be all 100% trans, which is true for all other 19 amino acids. For proline also it is 90% trans, only 10% can be cis. So what will happen if one proline is in cis rather than trans? So if this is the normal structure, let's say here is a proline and instead of being like this, it will be like this. That is cis. So and from this point onwards, the whole structure of the protein changes because it is in the wrong direction. So even though it is 10%, there may be 10 proteins in the sequence, because remember amino acids or proteins have hundreds of amino acids. So there can be 10 to 20 proteins in the whole protein. So eventually if one of them is cis, it gives rise to the wrong structure. And if you have so many proteins, then most likely the whole protein will have wrong activity. So even though it is 10%, you don't want even 1% to be in the wrong conformation, because that protein will not have activity. And if there are more proteins in the sequence, the chance of having inactive protein increases by the number of proteins you have. And that happens only with proline, and the cell cannot afford to have an inactive protein like that at random. And so there is an enzyme that corrects this. And that enzyme exists only for proline. It doesn't exist for any other amino acid that corrects the cis trans. So what will be the name of the enzyme? The name of the enzyme is based on what it does. So it takes a peptide bond, so it is peptidyl prolyl cis trans isomerase. So this enzyme scans the protein. Anytime it sees that a protein is cis, it breaks that bond and then reforms that bond. Remember, trans is more stable than the cis, so if the bond is reformed, it's most likely going to be a trans. And that's how the enzyme corrects any cis protein that exists in the protein. And only proline has this problem, so there is an enzyme only for proline. There is no such enzyme for any other amino acid.
Is this only between prolines?
No, wherever there is a proline, between proline and anything else, any other amino acid. Levels of organization in a protein, there are 4 levels of these three-dimensional structures. So first one is simply the primary sequence. And so it is called 1-degree structure, and so that is the primary sequence. So whatever is the sequence of the protein, so this is one amino acid linked to another one, linked to another one. So when you name those amino acids, like this is a glycine, then a tryptophan, then a serine, then a tyrosine. So when you write the sequence, that is the primary sequence. And that is the first degree of organization. And then you have secondary, which we already discussed. These are local structures arising in different regions of the protein that are stable, right? So that's how the alpha helix forms, that's how the beta sheet forms. So if you have a protein sequence like this, starting from here to here, let's say there are a couple of hundred amino acids. So from here to here, you may have a alpha helix. And then you may have a random coil here. Then you may have a beta sheet here. And somewhere again, the beta sheet cannot exist by one strand cannot exist, so somewhere else there must be a beta. So this way, and they may be interacting with each other later. So you have different regions that may have these different secondary structures. So the three-dimensional structure of the protein is not a straight line like this, but if this is the alpha helix, then it will be like this here, and then this is a random coil, and then there's the beta strand like this, and then again you may have an alpha helix if you want. So this way there are different regions, localized regions having different structures. And once again, all these structures must follow the Ramachandran diagram. They must follow the allowed angles. You cannot violate those angles. And then you have the tertiary structure. Which means the different secondary structures interact with each other. So the way I have drawn it is still a straight line with these secondary structures in between. But that's not the real structure of a protein. In a real structure, this alpha helix may interact with this alpha helix. So it will come back here. And as you know, the beta strands don't exist by themselves. They have to be associated with another beta strand to form a beta sheet. So that means these structures come together to form a three-dimensional structure like this, and that is the tertiary structure of the protein. So what is the stabilizing factor for a tertiary structure? Secondary structure we saw before, which is the hydrogen bonding for alpha helix, intra-strand hydrogen bonding for beta strand. So why do two beta strands come together? We already saw it come together because of hydrogen bonding. Why can two alpha helix come together? Again, all the intermolecular forces. So everything applies. You can have hydrogen bonding, you can have polar interaction, you can have hydrophobic interaction or non-polar interaction. So all those interactions bring this structure close to this one or this one, and they form a globular structure like this, and that is the tertiary structure of the protein. And then finally, there is a fourth one, which is the quaternary structure. So quaternary structure of a protein does not exist for all proteins. Only for some proteins, I shouldn't say some, but it is actually for majority proteins, it is about 80% of the proteins have quaternary structures, 20% don't. So quaternary structure is an interaction between two different proteins, two protein molecules. The two protein molecules can be from the same sequence, or they may have a different sequence. So two molecules of the same type may interact with each other, or two molecules of different type may interact with each other. In the same way, it may not be two, it may be three, it may be four, it may be six. So it is a cluster of proteins that stay together to form a quaternary structure. And as I said, about 20% of the proteins don't even have quaternary structure because they have only one protein strand that makes the whole protein. So if you have more than one, then each of these is called subunit. So quaternary structure will exist for those proteins that have subunits. And these subunits come together. There may be many reasons why they come together. One particular reason can be that the monomer may be less active than the dimer or the tetramer. That's why they are together. Some proteins are not active at all as a monomer. Only when they combine together they form the right structure to be active. So the reason varies from protein to protein. And the fact is that some proteins are monomers, some proteins are dimers, some are trimers, tetramers, like that. So what is the interaction between them? What force brings them together? Once again, all those forces that it can be hydrogen bonding, it can be polar interactions or non-polar interactions that bring them together. Yes.
I just have a quick question. So, for diseases like prion disease, which is like misfolded proteins, how would that, because we've been talking about how each proteins have to have certain like rules to make sure it's an active protein. So what goes wrong with like a prion?
You have brought up a topic for which the person who discovered prions, Stanley Prisner, wherever he went for giving a lecture, people just booed him. He said no one believed it. For so many years, everyone said this is all junk. And he lived with that for so many years. He loudly presented his work and no one believed it. So it's a very difficult topic, and your question is difficult also. Because this is the only example of a protein causing an infectious disease. And that's why people didn't believe it. People always knew that it must be a DNA that causes a disease. Infectious disease is always caused by an organism. It has to have DNA in it or RNA in it. It cannot be caused by a protein. And but then he turned out to be right, and he got the Nobel Prize for his work. So how it works? It works because one inactive protein induces another protein to follow that structure. Alright, so one protein somehow becomes inactive, has a different structure, and that induces other proteins to follow that structure, and that's how the whole, all the proteins become. So that's a crude way of explaining, but as I said, this started as a difficult topic that most scientists never even agreed to. And so giving you the exact answer to how that happens is still being studied. How exactly one protein can cause another protein to fold in the wrong way. So that brings us to this thing that we are talking about. As she said, this is called protein folding. Protein is not a straight line like this. It is a straight line when it is first made. Proteins are made in the ribosomes, and then when it is made, it is one straight line because it has to be made based on the gene. But then it has to fold to this final structure, the tertiary structure, and that includes interaction between this and this. So protein folding is a cooperative process. We will study many cooperative processes in biochemistry. What does it mean? It means the initiation of a process is difficult. That takes time. But when that initiates, the rest of the process happens much faster. So when proteins fold, remember it is one straight line, that's how it is made. But then at the end it has to be like this. What is the rate at which this converts to this one? This is a very short one I do. This is the long one. So this must convert to this structure. If you look at the rate, you will find that the first steps, that means forming this alpha helix, forming the beta sheets and beta strands, these are slow processes because, as I said, there are all these allowed angles. Out of that, you have to choose one angle and repeat it for every peptide bond, then only you form an alpha helix. So that process takes time. When I say takes time, it's not days or years, it is like seconds and minutes. Still, it takes time relative to other processes. But then once it is formed, it stays like that. Remember, a protein has trillions of conformations, really unlimited number of conformations. So in order to go from here to here, does it really look for all possible combinations of conformations and eventually get to this? That doesn't happen. It localized structure happens here, localized structure happens here, so every region a localized structure happens. Once it happens, it stays there. Right? Now this behaves as one unit. because this is already stable, that behaves as one unit. It doesn't separate again and start to look for another structure. So once a structure forms locally, that stays like that, and then this interacts with this one, that will be a very fast process, because all these 20 or 30 amino acids act as one unit, and that is looking for another 20, 30 amino acids which it can interact. So it's not one amino acid looking for other amino acid, it is a group of amino acids looking for other amino acids, so it folds rapidly. So that is called a cooperative process. That means it starts slowly and then goes faster. That applies to protein folding, as we will see. It applies to oxygen binding to hemoglobin. The first binding of oxygen to hemoglobin is slow. After it binds, then it rapidly binds more. So protein folding is also a cooperative process, but remember the final structure is the most stable structure. So it should form automatically. You don't need an enzyme to fold a protein. Protein should fold automatically. In fact, that was an experiment done by Ann Princeton, who got the Nobel Prize for demonstrating that proteins fold automatically. We are not going to study that. But basically the idea is he took a protein and then denatured it and then showed that it will form active protein back again without the help of any other enzyme. So protein should fold automatically because they are the most stable one. But then there is a problem going from here. This is coming out from the ribosome and then going into this structure. What is the problem? For which it actually needs help from other proteins. So there are proteins, not enzymes, there are proteins that help other proteins to fold. Even though the process should happen automatically by itself, that may take a long time to happen. So there are other proteins that help them to fold, and these proteins are called chaperones. Another name for the chaperones are heat shock proteins. In order to understand how the molecular chaperones work, you need to know how to make an omelette. Everyone makes an omelette, right? You start with egg, which is a liquid. You put it on hot frying pan, it becomes a solid. What kind of science is that? You always studied that you take a solid, you heat it, it becomes a liquid. There is an example where you take a liquid, you heat it, and it becomes a solid. Right? So anyone worried about that? We made so many omelettes and never thought, why is it becoming a solid when I heat it? That's the opposite. Yes? So here is what happens. So if you have a protein like this, they have all the side chains, hydrophilic, hydrophobic. So let's say there is a side chain here. I'm writing a C for all of them. They can be aromatic or any other hydrophobic group. So they are pointing both this. Of course, these are trans. So all the hydrophobic groups are pointing in both directions. In aqueous solution, this is an unstable structure. You cannot have hydrophobic group in water. So how will it become stable? It can become stable by combining with other proteins that also have their hydrophobic groups. So here is another protein that has the hydrophobic groups here. And so these point towards each other. And then another protein also like that, another protein like that. So all the hydrophobic groups are clustered together so that they stay away from the water. So instead of having one protein molecule, you have thousands of protein molecules. That increases the molecular weight, and that's why it becomes a solid. All right. So next time you eat an omelette, think about that. This is a cluster of many protein molecules, mainly albumin, that became a solid because all the hydrophobic groups have to come together. Now that is the problem for the ribosome also. Here is a ribosome that makes protein, so the protein is coming out. It will be just one linear chain, and then this has to fold. You have to give it time to fold. So what these molecular chaperones do, which are also called the heat shock proteins, they sequester each newly formed protein into a chamber and allows it to fold. So basically a molecular chaperone structure, if you look at the structure, it looks like this. It's a large protein, so this is a cylinder. So this is one subunit, another subunit. I think there are seven subunits of the protein together that forms a cylinder. So it is a cylindrical structure like this with a opening here. So these are the various subunits and on the other side also. And there is an opening here through which the newly formed protein is taken in. And so it doesn't interact with other proteins and it is given sufficient time to fold. Sufficient time means, again, in minutes, not hours or days, not also seconds, but it takes a couple of minutes for that to fold correctly. And so it gives that time to fold, and when it is folded, then it is released, and then another molecule of protein comes in. Yes? It separates it from other molecules of protein so that they don't interact with each other. And it basically gives it time to fold to the right structure. All right, so before we move on to the next chapter, as I said, another name for chaperone protein is the heat shock proteins. And that is because in science you will always see that different labs are working on different problems. And they are not in contact with each other also, and sometimes they meet and in seminars and conferences and they discuss their results, and it turns out that they are actually working on the same protein, same topic. So the people who work on protein folding, they realize that they are working on the same protein as people who are working on heat shock proteins. What are these heat shock proteins? If you take any bacteria and suddenly put them at high temperature, not 100 degrees, but let's say 45 degrees, which they don't die but survive, but it's a shock, so that's called a heat shock. So whenever bacteria are subjected to high temperature like that, they make certain proteins, and people were studying these proteins. What are these new proteins that the bacteria is making because of the sudden shock of heat? And it turns out later that they are the same as the chaperone protein. So that means these proteins are made in larger amounts because the bacteria was subjected to the heat, and it senses that many of its proteins will be killed or denatured, so it has to make more proteins and it has to fold those proteins. So these heat shock proteins are used for folding the new proteins which are now made in larger amounts. So that's the connection with bacteria people, or scientists working with bacteria and scientists working on eukaryotic cells. They were actually working on the same phenomenon, that is protein folding. All right, so as I said, there are the other protein that we are going to study is hemoglobin. And everyone knows the function of hemoglobin is what? It's like a transport company where carry oxygen, it's transporting oxygen. Just like you hire a transport company to transport your furniture, they are basically transporting oxygen. And when you hire a transport company, you want to make sure a couple of things, that they love your furniture, they don't want to just break it. So that means it must have affinity for what it is transporting. So the hemoglobin has affinity for oxygen. And then you don't want the transport company to love your furniture so much that they won't give it back to you, right? So you want them and deliver it to your new place. That's exactly what hemoglobin does too. It binds to oxygen, but then it doesn't bind it forever. It will bind to it and then it will deliver it. Where does it deliver? It delivers to the tissues. And it works like a transport company that is moving furniture for several different people, not just one person. And it will deliver it to the right address, the right number of furnitures. So hemoglobin does the same thing. It delivers oxygen to the different tissues, and it will deliver based on what the needs of that tissue is. All tissues don't need equal amount of oxygen. Some need more, some need less. For example, if you are running, your muscles need more oxygen than the other body that needs less. And the hemoglobin knows that. It knows which tissues will need more oxygen, it will deliver more oxygen there. And whichever needs less, it will deliver less oxygen. So that's a very intelligent function it has, that it will deliver the right amount of oxygen to the right tissues. Right? And after it has delivered, if the transport company comes back with an empty truck, that's a waste of money. They want to find another customer for transporting something else on their way back. Hemoglobin does the same thing. What does it transport on the way back? It transports carbon dioxide. Right? So that's not the only method of transport of carbon dioxide, but that is one of the methods of transporting carbon dioxide. So in fact, of all the oxygen, or of all the carbon dioxide that you breathe out, about half of it was brought there by hemoglobin. The other half was brought there by your blood plasma. Yes?
So does it help to control acidosis in that process? Like, because it carries the CO2 out of the tissue to the out of the body. Does it help with acidosis?
Yes, it does. Whether it controls acidosis or not, that's a different issue, the debatable one. But it depends, its function depends on the pH. So if it is carbon dioxide, as you know, dissolves in water to give carbonic acid, so it gives a lower pH. And the function of hemoglobin, whether it is transporting oxygen or transporting carbon dioxide, that depends on the pH. And as we will study later, that was discovered by, that is called the Bohr effect. Anyone heard of Bohr? Maybe you hear about Bohr. Chemistry, Niels Bohr. Bohr diagrams, all the electrons moving around, yeah, that's the Bohr also, but this Bohr is different. That was Niels Bohr, this is Christian Bohr. And it turns out that Christian Bohr is the father of Niels Bohr, so it runs in the family. Right? So Niels Bohr was a chemist or physicist. He looked at the structure of atoms, and Christian Bohr looked at hemoglobin. And he was the one who said, to answer your question, that transport of oxygen in blood depends on the pH. The pH affects the affinity of hemoglobin for oxygen. We will do that later in the chapter. So the hemoglobin, of course, is a protein, and the part of it that actually transports oxygen is called heme. Heme or hemin. So what heme has is a porphyrin ring, 4 of them, which will take me time to draw, so I don't want to draw that, but basically it is structured like this. It's all aromatic, and there's a nitrogen here. I send it to you by email. You should have it in the diagrams. And you open any book and look at the structure of the heme. And then there are 4 of them. then they are all aromatic. And then these are joined. It's not just 4 separated ones. These are joined by carbons like this to form this large structure. And in this large structure, which I have not drawn all the carbons, if you look at every double bond, it is double, single, double, then next one will be double, then next one will be single, next one will be double, like that. Throughout the ring, it is alternating double, single bond. And what property does that give? You have, this is called extended conjugation throughout the ring. So if you have extended conjugation throughout the ring, that helps it to have a absorption in the higher wavelength region. For example, if you may know that aromatic compound like benzene absorbs at 260 nanometers, which is UV range. If you extend those double bonds of the aromatic ring to the outside the ring, have more double bonds, one after another many, then from 260 it gradually moves on to the visible range. And that's how all these, anything that is colored is because of extended conjugation. For example, vitamin A has a color because it has many double bonds, and if you count them, they are alternating double single bonds. So because of these alternating double single bonds throughout the ring, this is colored. And what is the color? Not red. The red color is because of a different reason. The red color comes from iron at the center. And that gives it the intense red color that you have for blood. It is because of the Fe2+. So Fe2+ will have a coordination of 6. That means it will be bonded to 6 different electron donating groups. And so these nitrogens have electrons, so that coordinates this ion. And because the whole thing is planar, the whole thing is aromatic, this whole structure of heme is planar. Right? Aromatic things are planar, so the heme is planar. At the middle there is the ion, and it must be coordinated to 6 sources of electrons, 4 of which are coming from the nitrogens in the porphyrin ring. This is called the porphyrin ring. And then there are 2 others. Now this one is in part of hemoglobin. So this is a globin protein, let's say, globin protein. And somewhere is this structure with the iron in the center. And in the 3-dimensional structure, there happens to be a histidine just right below the iron. And histidine, remember, histidine has a side chain with a nitrogen and electrons. So somewhere below it, it's difficult to draw, but somewhere you can say at the back of this, below it there is a histidine. So the nitrogen of histidine is the 5th coordination. And then above it, what is the 6th one that binds to the iron? That is what we are here for, what are we studying? Oxygen, right? Transport to oxygen. Now there is a histidine here too. But that histidine is a little far away because there is space to bind. So I'm not writing the structure of histidine. It has a nitrogen in it, right? And that nitrogen doesn't really coordinate with this because it is close but not the right distance. It is a little far. So in between there is space for other molecules, and one of them is oxygen. So the oxygen coordinates with the iron to give you, that's how the heme is able to transport oxygen. So another molecule that, yes,
What's written on top?
histidine, there is another histidine there. The histidine is not the right distance for coordination, but it is very close. Alright? So in the absence of oxygen, that means when the blood is going back to the lungs from the tissues, what is there in the absence of oxygen? Not CO2. That was the guess. So what coordinates to the iron on its way back? It is the water molecule. So there is space for a water molecule to be here. So H2O in deoxygenated blood. So when it is transporting oxygen, it is oxygen, otherwise it is water. Now the reason I have this histidine, now remember this is a protein, so this whole thing is in the protein, so there is all these amino acids and somewhere there is a histidine. Right about this. The histidine is so close that if you draw a straight line from the iron, let me draw it this way, and here's the nitrogen of histidine. You don't have space enough for fitting in a molecule like oxygen. So oxygen will have to be at an angle which is actually its right structure. So from here to here, oxygen, and then here is the histidine that prevents any larger molecule to fit there. And not only that, if this oxygen was a straight line like this, in straight line, vertically straight from iron, that will not fit in there because the histidine will be obstructing. And that is actually good because oxygen has a bent structure like that. Why is oxygen at an angle and not straight? Because what is oxygen has a double bond, so what is the hybridization of oxygen? It is sp2. So that's why it has a structure like this. So it is the right angle for oxygen to bind. Binding always is because of the electron, so this electron binds to the iron and the oxygen will be like this. If you have a similar molecule that forms a straight line, then it will have less of a bind. Any other molecule you know that binds to hemoglobin very tightly.
Carbon monoxide.
Carbon monoxide. What is the structure of carbon monoxide? It has a triple bond. If you draw the Lewis electron dot structure of carbon monoxide, it is like this. It has a triple bond. So what is the hybridization? It is sp. So this is sp hybridized. Oxygen is sp2. So if carbon monoxide binds to hemoglobin, it will be a straight line from vertically straight from the iron, and there will be obstruction from the histidine there. So that decreases the affinity for of carbon monoxide for hemoglobin. But the affinity to begin with is so high, I don't remember the exact number, it is thousands of times higher affinity of carbon monoxide with the iron. And why is that? Because carbon monoxide has a full negative charge on the carbon. That makes it a higher affinity for hemoglobin. It binds to hemoglobin a couple of thousands of times more affinity than oxygen. But that affinity is actually slightly less because of the histidine that comes in the way, but still, even considering that, carbon monoxide still has a couple of 100 times more affinity than oxygen. If that histidine wasn't there, it would be a couple of thousand times. With that histidine, it is still a couple of 100 times stronger affinity than oxygen. And so even though the concentration of carbon monoxide in the air is extremely low, you still may get 1 or 2 molecules bind to it. In case of carbon monoxide poisoning, you don't have to have 100% carbon monoxide. Even at a slightly higher level of carbon monoxide, the person will die, because this is a very tight binding, and tight binding is not good, because as I said, your furniture company should not have too much affinity for your furniture, they will not deliver it otherwise. So anything that binds tightly don't come off. And so carbon monoxide, once it binds, it never comes off, and that's how the hemoglobin will be totally out of circulation. And that's how carbon monoxide can kill in a short time without even the person knowing it. All right? All right, so I think time is up. Right? We will stop. Have the place confused, and don't worry too much about the place.